welcome everyone to another part of BIS103 Lecture 7. In this video, we want to look at some of the really foundational concepts of the biosynthesis of poly and disaccharides. So our learning goal for this short video is that we want to be able to describe at least the basic chemical logic of the pathways on how we can biosynthesize disaccharides such as sucrose and lactose, as well as our polysaccharides, especially glycogen. Right? And it sort of accompanies our lecture on gluconeogenesis, where we talked about how we can go back from non-carbohydrate substrates to glucose. And we talked about that under certain circumstances, we can actually use that glucose and its precursor glucose 6-phosphate, not necessarily for generating chemical energy again, but to generate storage of carbohydrates in form of glycogen. So we just want to see, like, when we get to this point of glucose 6-phosphate, what actually happens, for example, under these circumstances when we want to make glycogen for storage. Just in simple terms, this is what is the first step here, right? This should look familiar. These are the amylose and amylopectin branches that we looked at when we talked about breaking down glycogen. And essentially, we're doing the same in reverse here. I remember your dietary glycogen was broken down by phosphorylasis. We use phosphate to cleave these glycosidic bonds here. If we actually want to make glycogen, we do essentially the reverse. What we're doing is actually we're bringing in your glucose here, right? And we're bringing in UDP glucose. Right? This should sound familiar from our lecture 4 when we talked about the utilization of other carbohydrates and specifically galactose. So have a look at that video, sort of remind yourself of how it worked when we wanted to bring in galactose into the glycolytic pathway. Right? We also used UDP glucose there. So again, right, UDP glucose, you have your glucose moiety here, and then you have this nucleotide type backbone, you have your base, your sugar, and then the phosphoanhydride bond. What we can do now is that we can actually use the cleavage of this molecule here to attach one glucose molecule in alpha 1,4 to the glycosidic bond here. And you're basically just elongating the branch one glucose molecule at the time at the reducing end. The next step then is, right, we had talked about glycogen being branched, and so is starch for that matter. Right, so we need an enzyme that does this. And this is simply called the branching enzymes. And I won't go into its mechanism here. Just remember its name and what it does. It will take these linear branches of glucose molecules and will generate these 1,6 glycosidic bonds to generate the branching within the glycogen molecule. That's all that I expect you to know for this enzyme. From glycogen, we can make other smaller disaccharides, for example. We're actually using the same strategy. So for example, cows in the milk that make a lot of lactose, right? Lactose is comprised of a molecule of glucose and a molecule of galactose. So in this case, we're bringing in one molecule of free glucose. And in this case, not UDP glucose, but UDP galactose. We're helping ourselves with the exact same mechanism. We're using the energy of the hydrolysis of our backbone here. And we are bringing about the closure of the glycosidic bond between galactose and glucose. We have generated lactose. UDP is being freed up, can be used again to capture another glucose molecule. You can do this reaction once again. How about sucrose, right? Our table sugar, usually derived from plants. How do plants do it? Sucrose is the major disaccharide found in plants. Similar strategy, slightly different. Here we're bringing in UDP glucose, and instead of free glucose, we're bringing in phosphorylated fructose, fructose 6-phosphate. Doing the same thing now, we're using the energy. We are actually now bringing in the glucose to the fructose 6-phosphate. which means we're not generating the disaccharide outright. We still have it phosphorylated right here. So we need one more enzymatic step here, which is a phosphatase, which we had seen in our lecture on gluconeogenesis. We're using water to break the group bond to the phosphoryl group, leaving us with the disaccharide sucrose here, made up of one molecule glucose, one molecule of fructose. This is just, in a nutshell, the very basic steps on how we can use glucose and our other sugar monosaccharide monomers to bring about the biosynthesis of disaccharides, as well as higher molecule polymers such as glycogen and starch.